Hi, I'm Tríona, and today I'm going to talk to you about two of my favourite things, food and science, and in particular we're going to talk about eggs and proteins. So to me, an egg is a nice little package that's actually full of little proteins floating around in there, and we can do nice things with them, like cook them. So what are proteins? Proteins are made up of amino acids. The DNA encodes what amino acids are going to go into the protein, so the cellular machinery reads along the DNA in 3-letter chunks, and each 3-letter chunk corresponds to a particular amino acid that's added to the chain. Now these amino acids have different properties. Some of them have positive charge, some of them have a negative charge. These would interact with each other, for example. Some of them are hydrophobic, which means they dislike water, and they tend to huddle together if they're in the protein. And these long strings of amino acids then are eventually made into like long structural proteins such as collagen, which you'd find in your hair or cartilage. But they can also be made into globular proteins, which, as the name implies, they're kind of globular. And what you'll find is that amino acids from one end of the chain and amino acids from the middle or the far end of the chain can now interact with each other. So the positive charge and the negative can interact. We'll have a little bundle of hydrophobic proteins hiding away from the water over this side. And these amino acids and their interactions are actually very sensitive to the environment around them. They can be affected by changes in the pH, changes in salt concentration, and, as we like to do in the kitchen, can be affected by changes in heat. So as you heat the proteins, you give it more energy. And these bonds that were previously kind of optimal bonds, you know, the positive and negative was good optimal state, all the hydrophobic lads together, that's an optimal state. They no longer need to be in the lowest energy. They can actually have all this extra energy coming from the heat. The protein can start to unravel, the bonds don't need to be there, and you end up with this kind of long entangled mess. But it's not happening in isolation. The protein next to it's doing the same, and amino acids from the inside that were once hidden away can now interact with other proteins, and what you'll find is that they'll cross-link, and you'll get this kind of a gel formed. And as we all know from boiling eggs, they don't tend to unboil, so there you go. This is what's happened when we've heated our proteins and totally untangled them into a mess. Now, we can use this to our advantage. If you measure the temperature at which egg whites set on average, it's about 60 to 65 degrees. And the average temperature that an egg yolk sets at is about 65 to 70. So we can see there's kind of two slightly different bands here. Now, if you cooked your egg at 60 to 65 and never let it get any higher, your yolk shouldn't really cook properly. It should just kind of start to solidify but not really go hard. But you'll have a perfectly set white, which is really important because most of the people I know, if you try and give them a wobbly white and a soft boiled egg, they'll freak out at you. It's just... So what we do is you can cook your egg at 65 degrees for up to an hour, and the egg yolk proteins, because they're 65 to 70, they shouldn't set as well. So you can end up with a perfect egg that keeps everyone happy and makes an absolutely tasty lunch.